Welcome again, friends, for the third lecture of our hematology series. This lecture will be discussing about origin, development, and morphology of blood cells. So let's dive into the amazing story of how our blood cells are made, which is a process called hematopoiesis or hemopoiesis. This is a continuous process. It is continuously producing inside your body in which the cellular constituents of blood are replenished as needed. So the meaning of this word meaning they will be produced when there is a requirement. So let's say there is some infection in your body. So white blood cells will be produced in more numbers to fight that infection. At the same time, if your body requires to clot to to make any clots, then also the platelets counts will be higher. So as per the requirement, hematopoiesis will occur in your body. Second point discusses about major three groups which we see in the blood. The first is erythrocytes, which are also called as red blood cells, leukocytes called as white blood cells, and platelets, which are also called as thrombocytes. Students, remembering these names are very important. The reason is sometimes in exam, or you can also guess in the government exam, this kind of questions can be asked directly from thrombocytes or erythrocytes or leukocytes. They may not directly say that red blood cells, they directly may not be give the description of the question about red blood cell. They may pronounce or write the erythrocytes. So we should understood the meaning of it. The whole blood cells are subdivided into major three categories. White blood cells, sorry. So major white blood cells we have already discussed in previous slide, which were neutrophil, eosinophil, basophil, monocyte, and lymphocytes. But out of these five, three are granulocytes: neutrophil, eosinophil, and basophil. These three considered as a granulocytes, then lymphocyte and monocytes are considered differently. So whenever you say, whenever you see 3-part cell counter, 5-part cell counter, or 6-part cell counter, the major difference is in 3-part cell counter. You will see that this granulocyte, this lymphocyte, and monocyte will be counted differently. In 5-part, all the major types which we have discussed previously, neutrophil, eosinophil, basophil, monocytes, and lymphocytes are separately calculated and counted. Okay, so basically blood cells will be originating from the specific location. It will not produce in the bloodstream. Okay, many of the students have this wrong idea about blood cells formation. So you should understand where our blood is formed. So it is forming in the specific location which is called as marrow, and this marrow is found inside almost all bones, and few of the blood forming organs also produce the blood cells. So let's look at where each type of the blood cells has been made and how many percentage of white blood cells are produced at which location. So basically all the red blood cells and platelets are formulated inside the bone marrow. 60 to 70% white blood cells are also formulated in the bone marrow. The remaining 20 to 30% of white blood cells are specifically produced in lymphocytes, which are also called as lymphocytes, which are produced in lymph nodes, spleen and thymus. These three organs are also called as lymphatic tissues. The reason behind calling it lymphatic tissues is it produces lymphocytes. The reticuloendothelial tissues of the spleen, liver, lymph nodes and other organs produce monocytes, which are about 4 to 8% of the white blood cells. Students, remembering the percentage or around total range, normal range of each of the white blood cells is very essential. I will prepare a different video for that, so you don't need to worry. The platelets, which are very small, which are also considered as a fragments of the cell, are formed from the cytoplasm of the giant cell, which is also called as megakaryocytes of the bone marrow. Now this is important because sometimes it is asked that the platelets are formed from which cell. So the answer will be the megakaryocytes. Figure, you can see it is showing the family of all the blood cells formulating from the single cell, which is called as multipotential hematopoietic stem cell. Now why it is called as multipotential? The reason is it can give rise to many of the cells such as thrombocytes, basophil, neutrophil, that means all the white blood cells, macrophages and other type of cells. So basically it consists of, or it may give rise to mainly two type of progenitor cells. The first is myeloid progenitor, second one will be the lymphoid progenitor. Myeloid progenitor will go under subsequent changes and produces a megakaryocyte, which is also called as giant cells, and once this cell is matured, it will broken down into the small fragments of the cells, which is called as platelets. Second, erythrocytes, RBCs, mast cells, myeloblast. Myeloblast is a stage which can give rise to multiple cells such as basophils, neutrophil, eosinophil and monocytes. Specifically, most of the white blood cells are formulated by the myeloblast. However, there is one lymphocyte which is missing from here, which will be formulated by this lymphoid progenitor. So monocyte will again give rise to the macrophages. Lymphoid progenitor gives rise to the natural killer cell and small lymphocytes. Now this small lymphocyte will again go under changes and produces T lymphocytes and B lymphocytes. B lymphocyte is also called as plasma cells, and it can give rise to formulation of antibodies. This figure also shows the similar kind of action made by the stem cells. Basically, two major progenitor cells are producing, which are also currently at the stage of stem cells, and they will give rise to the lymphoid and myeloid blast, and further they will produce a specific cell type based on the requirement. Hematopoiesis, which is the formation of blood cellular components, including red blood cell, white blood cell, platelets, and other type of cells. All cellular blood components are derived from hematopoietic stem cells. In a healthy adult person, approximately 10 raised to 11 to 10 raised to 12 new blood cells are produced daily in order to maintain steady state levels in peripheral circulation. So you can understand that how many types of blood cells are produced within a day. HSCs resides in the medulla of the bone marrow. So this medulla we will understand in the next slide, which is a very small portion but having a very soft tissue like structure. So all the blood cells have the unique ability, okay, to differentiated into the specific cell type. So you can say that a bone marrow is kind of a factory which is giving rise to a various products such as RBC, WBC types and the other types of cells. HSCs are self-renewing cells. When they differentiate, at least some of their daughter cells remain as HSCs. So the pool of stem cells is not depleted. So someone might get this kind of question that what if all the stem cells are differentiated into some other kind of cells? Then what happens? How new cells are produced? But our body and the creation of God is very smart and very complex. It understands the depletion, how can all the cells can be depleted. So it has the unique ability to again give rise to the new HSCs once few of the HSCs are utilized. This phenomena is called as asymmetric division, in which HSCs will give rise to the daughter HSCs. Hematopoietic stem cells, you can see in this figure, it is produced in yolk sac for the first few months, and then after certain period of time, the most of the amount of blood cells are produced in liver, spleen, and slowly after the fourth month of the child development, his body will produce slowly in the bone marrow, and then these different kind of bones are shown here, in which the maximum blood cells are produced in a vertebral and pelvis bone, which you can also see in this figure. It shows various bone types showing in various colors, in which vertebral and pelvis considered here, okay, in which the maximum part of the blood cells are produced. Here you can see in the y-axis it is a cellularity, and in the x-axis you can see is the time limit. As I was previously saying that the bone marrow looks like this. In most of your bones, there is a small portion which is called as the marrow, in which the hematopoietic stem cells resides, and this will give rise to the other type of cells. Maturation and growth factors. As a stem cell matures, it undergoes changes. So whenever any stem cells are maturing, that means it is undergoing certain changes. Changes meaning it is undergoing differentiation to make a different type of cell. Gene expression that limit the cell types that can become and moves it closer to a specific cells. So basically, what kind of cell HSC should make depends on the gene expression. Basically, all the things are written in the gene, and this will be decoded by the certain type of machineries, and then HSCs will decide whether to make which type of cell. These changes can be often tracked by monitoring and presence of proteins on the surface of the cell. So, as you know, the proteins on the surface of each cell is very different. Such as if we talk about RBC, the receptors of the RBC determine which type of blood it is. So, you know, even in the RBC, there are various kinds of RBCs are present. So, obviously, all the other types of blood cells have a different proteins on their surface. Each successive changes moves the cell closer to the final cell type. That means if our body requires to produce more platelets, then it will produce a specific cell type which can give rise to the platelets, such as megakaryocytes. So we can understand that our body currently requires formation of more platelets, and this is how we can track it. Red and white blood cells produced is closely regulated, and it is not only regulated, but it is regulated with the precision. That means our body should not make any mistakes making the blood types. Otherwise, it will be called as disorder. So, whenever there is a requirement of leukocytes in case of infection, our body will precisely produce more and more white blood cells. Then this type of cells will proliferate and they will go to the location where it is required by the blood circulation. So the proliferation and self-renewal of HSCs depend on growth factors. So there are certain growth factors which are important and necessary to give rise to this kind of specific cells. Hematopoietic growth factors are a family of regulatory molecules. Specifically, if we talk about in hematopoiesis, hematopoietic growth factors works very closely with the process of differentiation. They will give signals to the hematopoietic factory about which type of cells are currently required. As you know, as an example, if some person eats too much of the sugar, our body will indirectly signals the insulin to be produced. So similarly, the growth factors works accordingly as a regulatory molecules. So they will play important role in growth, survival and differentiation of blood progenitor cells. And they also function as a activation of mature cells. Erythropoietin is required for myeloid progenitor cell to become erythrocyte. So we have already talked about erythropoietin, which is a hormone which is closely related to the formation of the erythrocytes, meaning red blood cells. Thrombopoietin makes the regulation of megakaryocytes, which will give rise to the platelets. Okay, now let's see few questions, because as we now have understood the blood, its composition and its origin, development and maturation. Okay, so here are few questions that I want you to answer in comments. So first question is, which of the following blood cells play an important role in blood clotting? So here are few answers are written, but you can see that the terminologies which have used is different from red blood cell, white blood cell and platelets. So answer of this, it's very easy question. A will be the answer. Thrombocytes. Meaning of thrombocytes is also name given for the platelets. Neutrophils on the other hand, it is a kind of white blood cell, type of white blood cell. It is involved in the whenever a pathogen attacks, the neutrophil working as a granular molecule, discharge molecule cell and phagocytic cell. Next, leukocyte fighting infections erythrocytes, transporting nutrients, transporting oxygen, and other kind of activity. Okay. Okay, now this question is important. Which of the following is correct? The first option, serum contains blood and fibrinogen. Okay, now first of all, before going into the question, you should know what is the difference between serum and plasma. Okay, so just after telling the answer, I will explain you what is the difference between serum and plasma. Second option is plasma is blood without lymphocytes. Third option, blood comprises of plasma, RBC, WBC and platelets. This seems like right option, but never guess blindly. Uh, just select the option after reading all the options. The last option is lymph is plasma with RBC and WBC. So right answer is C. Why others are wrong answer? So first of all, serum contains blood and fibrinogen. So serum, first of all, do not contain blood because blood itself contains serum. Serum do not contain fibrinogen. So the first option is incorrect. Second option, plasma is blood without lymphocytes. So the wrong answer, it do not contain lymphocytes, but plasma is not blood because blood contains the plasma. D option, lymph is plasma with RBC. So lymph is not plasma and it is not RBC, WBC. So now let me explain you what is the difference between serum and plasma in upcoming slide. The third question states exactly what we need to learn. Serum differs from the blood as it lacks antibodies, clotting factors, albumin or globulins. So the right answer is clotting factors. Serum lacks the clotting factors. So first of all, let me explain you what is the difference between serum and plasma. So the serum is a liquid portion of the blood. Whenever we collect a whole blood from the patient, we can add it into two different type of vials, basically or majorly. First vial is having no anticoagulants. Second vial, what we can add is having the coagulants such as EDTA. So what happens in both of these vials? In first vial, as there are no anticoagulants, the blood will clot. In second vial, as there are anticoagulants which will stop blood from the clotting, they will be in the liquid form. So once the clotting has been done, that means all the clotting factors are utilized. So there will be no clotting factors present in the serum. The next thing, whenever we are using the EDTA like anticoagulants, then that means the clotting factors are chelated, but they are present inside the plasma. So whenever we centrifuge this both tube, from this no anticoagulant containing vial, we can get serum, and from this EDTA or anticoagulant containing vial, we can get is plasma. Okay, so this was the basic difference between serum and plasma. I hope I was able to clear this doubt. Okay, still if you do not understand, let me know. I will again explain in the upcoming sessions. So fourth question is plasma protein. Which of the plasma protein is responsible for blood coagulation? Fibrinogen, globulin, serum amylase or albumin? So here the answer will be the fibrinogen. Fibrinogen is also called as clotting factor, which is very important for clotting. So coagulation and clotting both are similar things. Okay, so whenever they use coagulation or clotting, it means the same. Okay, so the next question is an increased white blood cell count is indicative of which disease? The options are lupus, leukemia, anemia or melanoma. So right answer will be the leukemia. Why? Let's see one by one. Lupus is actually an autoimmune disease. It can sometimes cause low white blood cells, rather increasing it. So this out of the option. Anemia. Anemia is the condition in which there is a low red blood cell count, while not white blood cell count will be directly affected by the anemia. Next is melanoma. Melanoma is a kind of skin cancer, so it will directly not affect white blood cell levels. Okay, so the answer will be the leukemia. Leukemia itself is a cancer of white blood cells. So as you can see, the leukemia will cause the increased levels of WBC. Next question, very easy. Which of the following is function of white blood cell? Transporting oxygen, maintaining hemostasis, defend against infection, and produce hemoglobin. So the right answer will be defend against infection. So white blood cell transporting oxygen, it is of the RBCs, maintaining hemostasis, whole blood will do this, produce hemoglobin. So hemoglobin's production, we will see how the hemoglobin proteins are produced. So mostly it will be producing in the ribosomes, right? So the next question, platelets are formed from what type of cell? So melanocytes, macrophages, astrocytes or megakaryocytes. So we have just seen the answer in this lecture, which are megakaryocytes. Next question, the formation of blood clot is known as coagulation, chemotaxis, leukopoiesis or erythropoiesis. So answer we know is coagulation or blood clotting. Okay. Next question, where does hematopoiesis take place? Lungs, bone marrow, liver, pancreas. So as we have discussed, you have seen these both options in our slide, but always choose the bone marrow over any other options when there are no mentioning directly about any age or any certain criteria. Next question, which of the following statement about erythrocyte is correct? Fight infection, clot blood, lack nucleus, or they produce in spleen. So right answer will be they lack nucleus. As we have seen the structure, it lacks the nucleus.